Good morning to you all, learners and students. I am Professor S. Kripanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, located in a rural vicinity, Vatlamudi, Guntur district, Andhra Pradesh. And immunoinformatics. In this, we have covered so far 4 units. The first unit is on overview of the immune system, which involves more than 7 to 8 topics. The second unit was on immunoglobulins, wherein hybridoma technology also appeared, complements also appear. The third unit was antigen-antibody interaction. The fourth unit was on the T cell and B cell development. And the fifth unit in the course that is prescribed for the BTech students in biotechnology and bioinformatics is immunoinformatics, fifth unit. In this fifth unit, the first topic we covered, that is on immunoinformatics. The second topic is on epitope prediction. Okay, so that's what we are doing now. So in immunology, we had to deal with antigen and antibody, because antigen elicites immune response, and as a consequence of the immune response, antibodies are released. So as a result, antigen, antibody, both are consequential, one after the other. Okay, so whenever we speak of the antigen, antigen invariably goes with epitopes. Antigen is a foreign material. Foreign material means it's not from USA, it's not from, what is it called, Russia. So foreign means from the host. If you are human beings, for us the foreign is a protein from sporozoans, a protein from virus, a protein from invertebrates or insects, a protein from fungus constitute foreign. Phylogenetically distant protein is, we usually refer to as a foreign protein. Okay, so that's what the foreign, every protein is having certain molecular weight in Daltons. It is having a characteristic fold. Proteins are having characteristic fold. That's what the proteins are antigenic and immunogenic. Okay, it's not so with regard to the other DNA molecule or other lipid molecules. Okay, so whether it is having a characteristic fold, then there is a region which is said to be antigenic determinant sites, ADS. So that ADS are called epitopes. So a protein is having any number of epitopes, depending upon its complexity. If it is a big complex protein called immunogen, it will be having more like hemocyanin. If it is like bovine serum albumin, it will be having a few. It is like spike protein of coronavirus, it is having many. So like this, epitopes, antigen determinant sites are there. Now, so once you know the sequence of these proteins, we can predict on the computer which is an epitope. Which is an epitope. Sequential epitope, conformational epitopes, linear epitopes, okay, and again the T cell based epitopes, all those things. That's what we are saying. The epitope prediction, how is it possible? Through the various tools we are seeing now. So let us see the objectives. To understand the term epitope, and again let us know here consequential and conformational epitopes. To study the importance of epitopes in designing vaccines. See, ultimately we are studying all these 4 units of immunology, and again 5th unit we are studying how to design a vaccine. See, because vaccine for coronavirus will not come from elsewhere. It comes from the coronavirus proteins only, coronavirus genes only. Okay, so like this, how to make a vaccine from the pathogen, and to acquaint the web-based tools for epitope prediction. Let us see here. Expected outcome. The sequential and conformational epitopes will be known. The importance of epitopes in the design of vaccine will be known, because nowadays it is not with one epitope we do, multiple epitope based vaccines. Web-based tools such as epitome database and conformational epitope database, B-cell epitope database, and antigen antibody database will be known. So many things I will show you now. We will practice as a project work later. So this is a video on B cell development. Let us see.
Protein 3D is DNA STAR's application for exploring macromolecular structure, motion, and function. In Protein 3D, the analysis view displays results of any prediction methods that you choose to apply. One such prediction method, the B-cell epitopes neoclone method, makes it easy to predict linear B-cell epitopes for any protein sequence or structure. In this example, I've already opened a PDB file for a mutant form of the BRCA1 BRCT gene. Our eventual goal will be to view the solvent-accessible surface of the likely B-cell epitope in relation to the rest of the protein. So we'll start by applying a light blue solvent-accessible surface over the entire protein. To do this, I'll open the Surfaces panel and select Solvent Accessible Surface High Quality. And now I'll open the color bar and change that color to light blue. In preparation for applying the neoclone method, we next need to open the Analysis view by clicking on the Analysis tab. To apply the neoclone method, we'll go to the Methods panel on the right. This panel shows a list of Protein 3D analysis methods. We'll check the box next to B-cell epitopes neoclone. As the method is activated, the results from the antigenic and highly antigenic models are instantaneously displayed at the bottom of the analysis view. Antigenic predictions are light pink and highly antigenic predictions are dark pink. We can click the plus sign to the left of the synopsis to display individual graphs. A confidence score is assigned to each residue to indicate the probability of it being or not being part of an epitope. By default, residues with a confidence greater than or equal to 0.5 are predicted to be antigenic. That threshold can be changed from the parameters bar if desired. Here we'll leave it at the default setting of 0.5. Since an epitope is likely to be near the surface of a protein, we can get additional confirmation by applying the M&E surface probability method. As you can see, there's a large continuous section of sequence that is predicted to be near the surface and to have a high probability of being an epitope. I'm going to highlight this entire region and then change its solvent accessible surface color from light blue to pink. To apply that, so I'll go back to the Explorer panel, and now I'm going to fill that in bright pink. Now we can go back to the structure view and see a visualization of our putative B-cell epitope in pink. In the sequence view, note that our putative epitope selection has been underlined in pink to match the color we chose for its solvent accessible surface. For more information on the neoclone method, please go to the Protein 3D page of our website and click on the white paper entitled Precise Predictions of Linear B-Cell Epitopes in Protein 3D.
Okay, so we have gone through now the video on B-cell epitope wherein they have taken BRCA1 gene, BRCA1 protein, that is breast cancer associated antigen. Okay, so in that she has navigated through various steps and wherein she has shown through the pink color the dark color regions constitute the, what is it called, the antigen, high antigenic. Okay, so like that and again surface, surface antigens and all those things are shown here. It's better to go through this DNA STAR software tool that is shown here. Okay, let's go now. So what is epitope? Let us see the definition. So antigenic determinants, they are nothing but in simple terms if you want to say antigenic determinants. What is antigen? Antigen is antibody generator. Antibody generator is an antigen. Antigenic determinants or epitopes are the portions of the antigen molecule which are responsible for specificity of the antigen in antigen-antibody reactions. Whenever the antigen-antibody reactions that we are seeing, so that's what whenever you take a rapid blood test, if you provide our blood, if our blood is having coronavirus antigen called spike protein antigen, it binds to IgG of the rapid blood test kit. Okay, that's what the reaction, antigen-antibody reaction. As a result, color develops. A sort of enzymatic addition has been done so as to give the color after the reaction got over. They combine with the antigen binding site of antibody. That is antibody is having a Y-shaped structure that is deduced by Porter and Edelman in Nobel laureate. In this Y-shaped structure, the fourth region is called Fab. Fab means fragment antigen binding. Okay, to which they are complementary. So the epitope is complementary to the Fab sequences. In the Fab, there is hypervariable regions, 3 hypervariable regions. All these things we have seen in the second unit. So now, let us see the vaccine development in post-genomic era. That is reverse vaccinology. So in the diagram you see, in the top it is shown conventional vaccine development. In the top, conventional vaccine development. In that conventional vaccine development, the cultures of the microbe is shown. So the microbe is having several antigens in the top. So these antigens are being selected for the purpose of developing the immunoglobulins, and the vaccines are given as attenuated vaccine is given, or heat-killed microbes are being given, or inactivated microbes are being given as vaccines. That is a conventional procedure for which to prepare the microbe as attenuated microbe. It takes good many number of years, as it is shown nearly for 15 years. Instead, through the computer, through the desktop screen, we can browse the protein sequences, and from the protein sequences, as we have seen in the previous case also, epitope prediction. We can predict the epitopes as it is shown in the video, and that process takes about 1 to 2 years, so that we can ultimately synthesize the epitope sequences from the plasmids, wherein the DNA can be inserted, ligated. Okay, so that plasmid gives the epitope sequences that can be purified, and you can make the vaccine. So that's what it is, a reverse vaccinology. Reverse vaccinology with which we can make this vaccine at the earliest. That's what the informatic tools help in this line. So this is what again the vaccine is. The same thing I am putting in a colored way. We borrowed this from the net. And here the cultivate microorganisms is shown in the first one. This is what the conventional vaccinology, which takes 15 years. We have to cultivate, okay, then inactivate, then inject. Okay, in the inactivation process is called attenuation. Okay, attenuation and heat kill, all those things. Then the next thing is the sequences are now, all the sequences are there in DDBJ, DNA database of Japan, or even NCBA. So those sequences can be retrieved. Okay, if the sequences are there for a particular protein and corresponding the gene, and those sequences we can get it synthesized, and those genes can be cultivated in plasmids and plasmid vectors, and those plasmid vectors express now the protein, and the protein we can take, and this protein we can use as a epitope or a vaccine. That takes within 1 or 2 years we can prepare the vaccine. Antigen selection and validation. Okay, that's what the vaccine development. So now, global genomic approach to identify new vaccine candidate. So as we have been telling, the reverse vaccinology, genome sequences, later protein technologies, in silico analysis, and DNA microarrays. Okay, so here basically proteomics technologies and how to synthesize a protein from the oligonucleotide sequence that we cultivated in the plasmid, the expression vector, through the in silico analysis. Now high throughput cloning and expression. Okay, ultimately in vitro and in vivo assays for vaccine candidate identification. So like this is a regular procedure for vaccine candidate identification. Let us see next. Now the lowermost called gene and protein sequence database. Here in this slide, please see the lowest one, the gene protein sequence database. So this database you can get from NCBA or any DDBJ and EBA. So after having taken the protein sequence, now we are interested to develop the vaccine using spike protein of coronavirus. Okay, so then you have to get the sequence. The sequence, where do you get? So they are available in NCBA. Once it is there, then disease-related protein. So once several proteins are there in corona, more than 16, so we have to see the disease-related protein. So the spike protein attaches to the angiotensin converting enzyme. So that is the one which is making to have entry into the human lung epithelial cells. So that's what now this is related protein you have to select. So that needs to be blocked by our immunoglobulins. Now, after having taken the disease-related protein, epitope prediction here involves epitope prediction as we have seen in the video. Now, candidate epitope database we have to see, then vaccinome, then peptide, and multi-epitope vaccines we have to make. That's what in silico analysis. Next. Now, what are the types of the epitopes? The sequential and continuous epitopes. They are recognized by the T helper cells because T helper cell comes in all the immune, what is it called, discussions. T helper cell is the only one which will prompt B cells. B cell is the only one which produces the immunoglobulins. That is the ultimate immune response. Linear peptide fragments and amphipathic helical 9 to 12 MERS. Okay, so here amphipathic means both hydrophilic and hydrophobic residues. Then conformational and discontinuous epitopes. So it is recognized by both T helper cells and B cells. Conformation, both B cells. A non-linear discrete amino acid sequence, I will show you the next diagram, the next slides, come together due to folding. Because antigen is not a single linear thread. It is having folded pattern. And the folded patterns have different folds which are far apart by sequence wise, but they come because of the fold, they come closer. Those folds are called the conformational epitopes. They are nearly 15 to 22 residues. Properties of the epitopes. They occur on the surface of the protein and are more flexible than the rest of the protein because they are hydrophilic. They have the high degree of exposure to the solvent because of the hydrophilic. The amino acids making the epitopes are usually charged. That is, usually there will be, what is that called, glutamic acid, aspartic acid, arginine and lysine, even proline, tyrosine, they all will be there, charged and hydrophilic. So now methods to identify epitopes. Usually immunochemical methods we adopt like ELISA, immunofluorescence, radioimmunoassay. Basically ELISA and immunofluorescence we do. And X-ray crystallography structures, it is a little bit costly affair. Antigen antibody complex has to be done. It is not there available in all laboratories. We can send the sample for this analysis. Okay, we will be ultimately getting the PDB structure. So prediction methods based on the X-ray crystal data available for antigen antibody complexes, the propensity of an amino acid to lie in an epitope is calculated. See, if we take some of the amino acids, nearly 600 amino acids will be there. All 600 amino acids cannot be having a high prediction for an epitope. The propensity of an amino acid towards an epitope can be predicted through these software tools as it has been shown in the video. Now, antigen antibody complexes, they involve proteins that must also exist independently. Remarkable features of antigen antibody complex are high affinity and strict specificity of antibody for their antigens. That's what everywhere for the detection we go for immunoglobulins. Immunoglobulins are having high specificity, high sensitivity towards the epitopes. Antibody recognizes the unique conformations and spatial locations of epitopes and surface of the antigen, because antibody is made against the epitope, so it can go its counterpart where the epitope is there on the conformations present on the protein. Epitopes and paratopes are relational entities. Okay. Now the first one is antigen antibody complex, wherein the blue and green shown is the antibody, the red one that is shown is an antigen. Here the second one is MSC with peptide. This is MSC molecule wherein the peptide is placed in the center. The peptide is anchored. Here again the MSC epitopes and peptide epitopes is again a big issue with regard to the computational predictions. So now see the sequential and conformational epitopes now. We'll be having a good understanding in this slide. So this particular immunoglobulin structure, Y-shaped structure, deduced by Port and Edelman Noble arrays. We are discussed in the second unit where there is Fab, where there is a FC region, crystallizable region. Fab is the region where antigen binding is shown as antigen binding site. Okay, so this is the region where antigen gets bound, antigen binding site. It clearly is shown. Now let us see. Now in this case, our interest is to get to know the sequential and conformational epitopes. Now let us see. The green one that is shown, it is an antibody, Y-shaped forked one. So here in this, there's another antigen that is getting bound. That antigen is sequential. It's a line. It's a green line is shown. Green line with the blue, it is shown here it is a, what is it called? Continuous epitope. Sequential epitopes. Contiguous epitope. Okay, so the epitopes here are both maybe hydrophilic, phobic, hydrophilic and hydrophobic, and hydrophilic ones are getting anchored to the fabric. That's what here sequential, contiguous. Now you see the other immunoglobulin which is there in red color. Okay, so here in the Fab region, see please in the Fab region, there are two of the antigen folds. So the two folds are not continuous. Okay, one fold is emanated out of the folding pattern that has become epitope. The other fold is emanated out of the folding pattern at far off distance that has come here because of the folding. Folding made it to come together here. So these two are sequentially apart, but they are together towards for the antibody to recognize these epitopes in the Fab region. This is what's called conformational epitopes or discontinuous epitopes. Okay, so that is the meaning. You have to remember this continuous and discontinuous epitope. This discontinuous epitopes is nothing but conformational epitopes. So this two prediction is most important. Sequential and conformation, antibody binding site. These are the antibody that binds them. The paratope of the antibody binds to the epitope of the antigen. So there are several algorithms. All these algorithms have been worked out by computational biologists. We are only the users of this. Okay, now this computational epitope server is there in the University of Pune, and it predicts the conformational epitopes from X-ray crystals of antigen-antibody complexes. It uses percent accessible surface area. Okay, now if there is a protein is having hidden area and surface area. So within the surface area, the accessible surface area and distance as criteria. Accessible surface area. Please remember. So that's what it uses for CEP, conformational epitope prediction. So these are all the various algorithms involved here. If you go through the residues, the second one, the residues having the down one, the residues having ASA. Okay, that is percent accessible surface area, 25% are termed as accessible residues. From that accessible residues, they take antigen determinant. Okay, so like this, all these things are being followed by computational biologists. Ultimately, a list of conformational epitopes is provided through HTML format. So now let us see a few of this epitome database. This is the database structurally already prevailing, structurally inferred antigenic epitopes. They're all, it is a big resource for that. And again, a brief introduction to CED, that is Conformational Epitope Database, is also available. The URL is given. And again, B-cell epitope prediction, wherein a database is available. And here again, antigen-antibody database is a home page, wherein the interaction of these two with regard to the van der Waals forces, hydrogen bonds and interactions and salt bridges, okay, and peptide bonds, all those things we'll be seeing here, the interaction. And again, let us see the graphical view of genome organization. Here we have taken a viral, what is it called, the genome, wherein the graphical view generated dynamically using scalable vector graphics technology, genome view. So here multiple sequence alignment we have to do, and through that we'll be knowing which is the signature sequence with which we'll go for the prediction of the epitopes. So it is the browsing the module of whole genome phylogenetic trees. So this is the most parsimonious tree of genus Flavivirus. Okay, parsimonious means it is very minimal by which we can determine the Flavivirus. So here let us see a simple example of mumps virus, antigenic diversity of mumps virus, an insight from predicted 3D structure of hemagglutinin neuraminidase protein. Okay, let us see now. So here the viral gene database. In this database, see here there is F called the fusion protein. F is there, the blue one. F is there, and again there is HN is there. F and HN is there. That is hemagglutinin neuraminidase. So this is the genome sequence. From there, the protein sequence have come. Okay, this is hemagglutinin neuraminidase predicted protein HN. So here in you can see the helical structures, which are red in color shown, and beta pleated sheets are shown in the yellow colors, and coils, random coils are shown here. Okay. Now, so mapping of mutations. So this virus keep undergoing the mutations. So that's what the first one and the other three ones, if you go through, they have shown in colored what is that called implication, where this mutations have occurred. Okay. So with this, let us see the definition of epitope, and figures depicting the sequential and conformational epitopes are shown in summary. Types of epitopes and their properties are shown. The reverse vaccinology in the prediction of epitopes is shown. Methods to identify epitopes and software tool databases are shown. The typical example of mumps virus genome and its antigen, namely fusion protein, hemagglutinin neuraminidase, with the mapping of mutations on 3D structure is shown. Okay, let's have the study questions. Define the term epitope. What are sequential and conformational epitopes? That's what we are to begin with. We have to start there. Indicate properties of epitopes. What is reverse vaccinology? How is it different from classical vaccine procedures? Indicate methods to identify epitopes and software tools database. Okay, and what are F and HN genes in mumps virus? In which context they are discussed in this particular topic? Okay, with this, let us acknowledge online resources and public domains from which we borrowed the information to develop the contents of teaching material. Thank you, Vananda.